I'm just going to go back to something you mentioned a little bit earlier, which was around ensiling time and the, starch digestibility. And the question in it is whether you have any insight from a genetic side of things, flint versus dent hybrids. So, in Australia, we've got a bit of a mix ranging from tropical genetics, temperate genetics. We've got really, really soft dent, and then we've probably got semi-dent and probably very, not a whole bunch of flint. Just trying to get some thoughts from you, Bill, around does it have any impact, flint versus dent versus semi-dent type hybrid in terms of starch digestibility?
Yeah, and we've done a lot of work in this area because Pioneer is really the first multinational company that has introduced dent into the Maritimes in Europe, where everything pretty much was flint before. So we've done a lot of work at the University of Paris. We've done work in Germany, where we've actually done trials to look at this topic. The difference between flint and dents really is flints are like popcorn. So it's a very hard vitreous starch, really encased in a lot of zein proteins. So very much more difficult for rumen bacteria to access those starch granules. Whereas dent corn has more of a floury endosperm, especially at silage harvest maturities, what the kernel is at silage harvest. Now, if we were to let grain go all the way to combining maturity, where we'd have it as dry corn, then we can have hybrids that have more of the hard starch, the vitreous starch, kind of like popcorn, on the outside. And that's what gives a hybrid higher test weight. Okay, but all of these, will digest differently in the rumen depending on how long it's been fermented, for example. Dry corn is an entirely different subject, but flint corn versus dent corn, after it's been in the bunker for about 6 months, they both feed pretty similarly, even before 6 months, maybe 4 months. They both feed pretty similarly. One of the issues is people only talk about ruminal starch digestion. And yes, we know that we want to have enough starch to grow rumen bacteria because they're a great protein source. Well, we don't want to feed so much starch as we cause acidosis and cause our cows to yield less components. But what I think many seed companies, not Pioneer, but many seed companies, when they talk about this digestibility, they only talk about the rumen because we have a laboratory analysis now called the 7-hour ruminal starch digestion. But people forget that the word ruminal is in there. Okay, that doesn't mean even with flint corn, maybe it's a little less digested in the rumen than a dent corn would be, but both dent and flint corn is going to be digested in the small intestine, in the duodenum, the first part of the small intestine, as long as it has the right surface area available to it. Now, if it's put up with a chopper and the kernels aren't processed correctly, it doesn't matter what kind of genetics, it's going to end up in the manure. But again, it's kind of a complicated subject to get into, is looking at ruminal starch versus post-ruminal starch. And one of the advantages of corn in your environment, as I see it, compared to the barley and wheat that you're feeding, is barley and wheat, because it doesn't have all that protein or as much protein surrounding the starch granules, and the starch granules are more loosely contained in barley and wheat. That's going to be more rapidly digested in the rumen. One of the advantages of corn, I think, in your diets is that some of that starch is going to get down to the intestine. So we're kind of protecting the rumen. We don't have this acidosis going on. And energetically, the cow gets more megajoules of energy from corn when it's digested in the intestine than it is in the rumen. Because in the rumen, it's got to go to propionate. Propionate's got to go to the liver. We got to convert it to glucose, and that's what the mammary uses. But when it goes into the intestines, boom, it goes right into as glucose. So there's a lot of nuances about it. The big advantage of dent corn over flint corn, it just yields so much more. All you got to do is look at the little kernels and no kernel depth to flint corn, and dent corn just yields so much more. There is no flint corn in North America, none, except Indian corn and popcorn. Again, it's just because it yields so low. You might say, well, why did the Maritimes, why do they even have flint corn? Well, flint corn historically has had pretty good early vigor. It jumps out of cold maritime soils a little bit better than dent corn did. But today we've got dent hybrids that have just as good early vigor. And one of the things I learned from our breeders in France in particular, where we've introduced a lot of dent hybrids, is France has recently gone through some droughty conditions. They saw, wow, these dent hybrids do so much better in drought than the flints. And here's what the breeder told me, is dent hybrids will tend to push roots first, vegetation second. But if you're a farmer, what do you want to see when you plant? You want to see vegetation. You can't see the roots, okay? But when the roots are going first, okay, that's going to be a lot better if we get into a drought situation. And so but today we have dent hybrids that have just as good early vigor as Flints do. And then I think in the next 10 years, and even some of the national companies in Europe are starting to introduce dents because they just yield so much more.
There's a lot in that, Bill. I think we might have to have a beer and explore that a bit further, I think, when you're over here. But yeah, so a bit of a trend that we've seen here in Australia in the last probably 10 years is we're starting to really see a lot more stay green in our hybrids, and that's allowing us to push our starch later. I'm talking in a silage situation here, and going closer to brown layer or even black layer on the cob. Is that actually helping us? Is that giving us a better feed, or are we better off to chop a bit earlier?
No, absolutely. And in the areas in the states where we're not using BMR because they just can't find enough acres, what they are really focused on is this late season plant health. I don't like the term stay green, never have for 30 years, and we're getting ready to excise that out of our vocabulary, I hope, in North America. We much prefer the term late season plant health, because stay green is really determined by breeders at combining maturity. When the plant is way past black layer, they'll look at how many of the lower leaves are fired up and put relative score on it. Our breeders are starting to look at a new score where we will actually look at what the late season plant health is at, say, 3/4 milk line, or, just before black layer, and have a new metric that we can put our hat on there. But again, the thing about the late season plant health that's so important is it'll help us maintain that fiber digestibility. And we've got over 127,000 data points, different silage analyses, that if the late season plant health is good, fiber digestibility in corn really does not decline very much at all. But what it does allow you is every day you keep it in the field, you can lay down upwards of 1 full percentage unit of starch per ton. To give you an idea what that means, every day we leave it in the field here in North America, we can lay down about 5 bushels of grain in that acre per day. So it'd be like shutting off the lactation of a dairy cow after 200 days, saying, well, okay, she's done. Okay, instead of gaining all that marginal milk that's left over. Same thing with a corn plant. The starch deposition is not linear, it's curvilinear, and we don't want to shut that off too soon. So if that plant stays healthy later in maturity, and we can go further out, as long as we can maintain that fiber digestibility. If we start to lose plant health, we're going to have to go in and chop earlier. Because here's how a nutritionist thinks about it, and why a lot of nutritionists are saying pulling the trigger maybe a little bit early. They're saying I can always add starch if I have to into the diet. I cannot add fiber digestibility. So they're kind of always erring on the safe side there. But I think we've proven to at least nutritionists here in North America that, in healthy plants, we can let that go. And in fact, I was just talking to some folks in Idaho. They're going all the way to black layer. There's a starch deficit state. They go all the way to black layer. That plant's still green, still very, very healthy. So why not take as much starch as possible? And as long as we get a good job of kernel processing on the chopper, we're good to go. So yeah, this late season plant health is critical. Now we have a lot of foliar diseases. I don't think, Ben, you told me you don't have quite as big an issue. Some northern maybe, or?
Yeah, probably not broadly. And I think probably our biggest dairy state in Victoria, it's not such a big issue. We don't see such big NLB, northern leaf blight challenges, or even rust and other things. It really is probably just that general late season plant health.
And I guess it's the biggest issue we have I stop saying stay green, but it's probably two-spotted mite. So I guess it impacts the greenness of the crop, but it's not a plant health type issue.
But I would suspect, though, that the fiber digestibility under that stress, we get the same thing with spider mites. We'll lose fiber digestibility with spider mites. Are we talking the same insect?
Yep.
Okay. The other thing we have is a lot of foliar diseases, and one that's just devastating is called tar spot, and it'll kill a plant in 5 days. In addition to breeding genetics for late season plant health, we also, almost as a routine management practice, are using fungicides or insecticides for miticides for spider mites. So we do some of those sort of things to help maintain that fiber digestibility and starch. I just learned something the other day. You learn something new every day. I had a breeder tell me this, that 50% of the photosynthate produced in that plant is the ear leaf. Another 20% is the leaf above it. If we lose those leaves, we've lost a lot in terms of being able to lay down starch. And so, again, that's why we're really trying to really protect these as much as possible.
Yeah, it's definitely more of a trend here now to deal with spider mite or two-spotted mite. It's probably in the past was seemed to be a bit difficult, but now I guess growers are more aware of it. It does become an issue for us in hot environments, for sure.
Another way, Bill, of I guess trying to manage that fiber digestibility side of things. I mean, we have people over here playing around with chop height to raise your chop height. What can you tell us a bit about that and the impact, positive or not, that it can have on the fiber digestibility?
It can have a pretty significant effect, but it really depends on the growing season. We've done a lot of work in this area, tremendous amount of work. And people kind of forgot about it once BMR was commercialized. But where you get out west where they're not using BMR, there is still a lot of high chopping that's done because the lower internodes are the least digestible portion of the plant. And if you high chop, you can also concentrate the starch. So you can find where, on paper, looking at a lab analysis, you can find where high chopping looks on paper just like it was BMR in terms of the 30-hour fiber digestibility value. But it won't feed in the cow the same way. That's a really important thing we have to understand, is because in a laboratory analysis, whether you're using NIR or wet chemistry, the NIR is based upon the wet chemistry. But think about it. We put the sample in a test tube. We put a buffer in there because the test tube isn't chewing your cud, producing saliva. And then we put rumen fluid in there, and we let the bugs have at it for 30 hours. Okay, and then we could do the same thing with BMR. Take them out. We've got two fiber digestibilities that may look exactly the same, but in a test tube, the feed can't go anywhere. But in a dairy cow's rumen, it can. And BMR exits the rumen much quicker. And so even though on paper, many people say, well, high chop, wow, I got the same numbers as BMR. It will not act in the cow the same way. They will be different. You'll get more energy out of the high chop for sure, but it won't drive intakes. All the research with BMR that's been done, all of it shows almost the same results, where you're feeding half to 3/4 of the forage, not as much as I'm saying 80% like you see today. Most of the research was done years ago, but where, say half of the forage was corn silage. When it was BMR, they were driving about 1 kilo more dry matter intake per cow per day. 1 kilo more intakes, about 4 pounds more milk. That's really the whole reason why BMR will drive intakes. So we have to be cautious. High chopping can work. It works more on irrigated ground than it does dry land ground, because irrigated corn's always lower in digestibility than dry land corn, just because of the rapid growth and that we get out of irrigated corn. So yeah, I'll show some data when I get over there. But again, it looks on paper like it almost makes it feed like BMR, but you got to watch the numbers.
Probably, I guess, the last thing as far as silage management and fiber digestibility, preserving the feed after you've got it out of the field. So, Pioneer's done a lot of work over the past on inoculants and that preservation side of things of silage. What can you tell us a little bit about? So over here we've got the couple of Rapid React products and some NutriVale. Can you talk us through a little bit about how they can be or should be used as well in that preservation piece of your silage stack, and then going through to feeding it out? Yeah,
I mean, when I first started with this company 39 years ago, that's what I did. I came in as a silage inoculant person. What I'm so proud of is Owen Newland was former vice president of Pioneer. He went and bought a company out of Portland, Oregon, that was producing silage inoculants, because what he said, which really makes me proud to work for this company, is that it does us no good to put up the best genetics in the world if we don't get it to the mouth of the animal. You guys may not even realize that Henry Wallace, the founder of Pioneer, and if you've been on our campus here in Iowa, he milked 30 cows. He was a dairy farmer before he was a corn breeder. his degrees from Iowa State University are in animal science, not plant science. And so we take our history of Pioneer is really based around livestock as well. In fact, up until the early 70s, Pioneer in the United States, we sold beef bulls and beef semen. Most people don't realize that. And also, Henry Wallace's son started Highline Chicks, which is the foundation breeding stock for layers in the whole world. Iowa is the #1 state for egg production, in addition to corn and soybean production. And that was started by Henry Wallace's son. And so we have quite a history for knowing that our products end up in a diet somewhere of an animal. And that's what we really pride ourselves on with our inoculants. And no other company has done this. Our inoculants are proven not just in a test tube in the laboratory, but being fed to animals. We have got research data where it's fed to animals to see what kind of response we can get. So we know a lot about preserving the sugar, keeping the sugars and more efficiently using those sugars so that there's more left over for the animal. And the NutriVail 11-CFT is an unbelievable product. This year in Florida in July, United States is going to be hosting the International Silage Conference. Back in 2018, it was in Bonn, Germany. The lead speaker for the conference in Bonn, Germany, was a researcher from the U.S. Dairy Forage Research Center named Dr. Richard Muck. And in his speech that he gave to kick off the conference, he mentioned one of the most innovative things that happened in the silage world was a particular silage inoculant that contained a Buchneri strain that produced ferulate esterase enzyme. That's our product, 11-CFT. And what's so interesting about it is many people talk about having, enzymes in their products. Well, you couldn't put enough enzyme in a product to affect a ton of corn silage. Impossible. You just don't have enough enzymes to be able to do that. But if you could find a bacteria that grows in the silage mass to 1 billion counts, and then that is producing an enzyme, that's really cool. And that's what 11-CFT is. And the big thing about it is people don't understand that a normal forage analysis just won't show that it did anything. We have to do a more sophisticated analysis that shows the rate of digestion. It changes the rate of digestion. So just looking at a single time point, like a 30-hour fiber digestibility, won't indicate what that product can do. So one of the things I will be introducing is a laboratory analysis called Fermentrix. In fact, I just had a conversation with Dr. Ian Lean. He is actually in Canada where this analysis is being performed. He's quite familiar, very aware of it, and I'm excited to work with him to teach you guys how to use something like this that can really help refine both looking at the rates of fiber and starches in diets, TMRs, PMRs, but also it can really show you how a product like the NutriVail 11-CFT can change the rate of fiber digestion. And it's just an amazing product. It hasn't taken off the way I would have hoped, only because it's a more sophisticated sale. We have to really work with nutritionists to understand what we're talking about when we talk about KDs and rate of digestion and that sort of thing. But hopefully that's something we'll be able to share with the growers in Australia when I'm over there.
Like and follow us for more episodes like this one.